Thank you very much, Simon, for the introduction and for the invitation to be here today. I actually changed my title, but I promise I will be talking about the genetics of early human embryos at the end. So, well, I have nothing to disclose. So, the main aim of the group, of Magdalena Sernica-Getz group, is to understand why so many embryos fail during the early stages of post-implantation development. This is very nicely represented here in the pregnancy loss iceberg. You can see that up to 30% of the human embryos fail from day 7 to day 14. So why is this the case? Of course, this is a very complicated question because now we have two players, the embryo and the endometrium. And as Professor Simon very nicely introduced the endometrium, I will be focusing mainly on the embryo. So why so many embryos are failing? What we know is that in 85% of the cases of pregnancy loss, we have fetal abnormalities. And in 50% of the cases, we have chromosomal abnormalities. So clearly, aneuploidy is a major factor that induces pregnancy loss, but is not the only one. But before we really address why so many embryos are failing at these stages, we thought it would be important to understand what are the morphogenetic transformations that a normal human embryo needs to undergo at these stages, just in normal conditions. But first of all, how could we study that? How can we study human embryo development beyond implantation, beyond day 7? So this was pioneered in the last century by the Carnegie Institution of Washington. They collected more than 10,000 human embryos and described their development. These were embryos that were developing in vivo in the uterus, and they were obtained through voluntary hysterectomy. So you can see images here from these publications. They collected embryos from the 2-cell stage all the way up to gastrulation. But now, to move on, which other techniques could we apply nowadays to study human embryos at these stages? So we know that we can co-culture human embryos with endometrial cells, and this is a really great system to understand the crosstalk between the uterus and the embryo. But whether the embryo undergoes normal morphogenesis in this type of cultures has not been addressed. So we thought if we want to develop a system where the embryo undergoes normal morphogenesis, we need to recapitulate this very complex transformation. I'm showing you here an image of an in vivo developing embryo, day 6 blastocyst, and how it looks some days later. It completely changed the shape and morphology. So we know that during these few days, there are lineage specification events. We have the 3 main tissues specified, the embryonic epiblast tissue and the 2 extra-embryonic tissues, hypoblast and trophoblast. But also we have morphogenetic movements and transformations. The epiblast forms the amniotic cavity, the hypoblast forms the yolk sac, and the trophoblast differentiates into cytotrophoblast and syncytiotrophoblast. So our aim was to develop a system that would allow us to study all of those transformations in vitro and beyond day 7. And we thought to do it just from the point of view of the embryo, to develop a system where we only have embryos, so we don't have the confounding effects of the endometrial tissue. And if we could do that, that would mean that the embryos display self-organizing capabilities, meaning that they can undergo morphogenesis on their own. And we were in a perfect position to address this question because the laboratory of Magdalena Sernica-Getz had a lot of experience throughout the years on how to achieve that in the mouse embryo. So here you can see a pre-implantation mouse blastocyst that we can culture in vitro in a dish, no endometrial factors. This embryo develops all the way up to post-implantation to form this cylindrical structure that is characteristic of post-implantation mouse embryos. So we applied the same kind of method to human embryos, and we observed that the human embryos were able to survive in this culture method and they attach. But now, are they undergoing normal morphogenesis? So these are just some drawings to show you the type of structures that we want to analyze. So here you can see that this embryonic tissue, the epiblast that I'm showing in yellow, forms the amniotic cavity, becomes epithelial. The hypoblast forms the yolk sac, and the trophoblast differentiates. And this is just to have the comparison of how the embryo would look like later on during the pregnancy. So first of all, we could see that the embryos were growing throughout the culture from day 7 to day 11. They grew on size, the number of the cells also increased. And then we observed also lineage specification events. So you can see that now we have both the epiblast tissue marked by the OCT4 expression and the hypoblast tissue marked by GATA6 expression. So the inner cell mass had separated into these two main tissues. And the separation was maintained throughout development. This is a later embryo, it's a dated embryo, and you can see here the epiblast compartment and the hypoblast cells. Another major milestone was amniotic cavity formation, and this is an embryo that has just initiated this process. So you can see here this polarized localization of this apical polarity protein, APKC, just at the site where the cavity is just formed. So you can see this very small cavity at day 8, but again it will grow in size. This is the 3D reconstruction of a day 10 embryo, and you can see the cavity in red is now much bigger. And also importantly for us, we can recapitulate this process with stem cells. This is important because it allows us to do functional studies. So by culturing human embryonic stem cells in a matrix, 3D matrix, we can reproduce this amniotic cavitation. So actually, by doing these experiments, we realized that mice and humans are not that different, at least at the beginning. So one of these embryos is a mouse, one is a human. And I'm sure most of you would not know which one is the mouse, which one is the human. So you see they are very, very similar, and this initial transformation to make the amniotic cavity happens in a very similar manner. And we call this structure, this organized structure, a rosette, where all the cells are organized around this incipient cavity. But then we know that later on in development they will have completely different morphologies. So here you can see a mouse post-implantation embryo with the typical cylindrical shape, and this would be a human embryo with the disc shape that we have cultured in vitro. And then we also observe the formation of the yolk sac that I mentioned is derived from the hypoblast. So you can see here these GATA6 positive cells that are surrounding this second cavity, the yolk sac cavity. And finally, we observe differentiation of the trophoblast into the two main cell types, cytotrophoblast and syncytiotrophoblast. So the cytotrophoblast are these uninucleated cells. This is the stem cell population of the trophoblast. And the syncytio are these multinucleated cells that we see at the periphery of the embryo. And actually, these findings were also observed in the group of Ali Van Loo, and they utilize specific markers to label these different populations. So you can see here GATA3, a marker of cytotrophoblast, expressed in these in vitro culture embryos at different stages, and then HCG, marker of the syncytiotrophoblast, also expressed in these in vitro culture embryos. So in summary, we have been able to culture human embryos beyond the implantation barrier. And by doing that, we have learned that human embryos have this remarkable self-organizing capacity. They are able to do all these beautiful transformations in the absence of maternal tissues. And we have also learned that in both mouse and human embryos, the epiblast undergoes this process of epithelial tissue formation by making a rosette. But now what can we do with this system? And going back to the beginning of my talk, I think this is a great system now to study the possible mechanisms of early pregnancy loss. So I will be telling you a story about how the embryonic tissue undergoes these first morphogenetic events, and then I will discuss a bit the role of aneuploidy. So we thought, thinking about embryonic morphogenesis, as I already mentioned, the first thing that these embryonic cells have to do when the embryo implants is to become epithelial and form this cavity, the amniotic cavity. So the hypothesis would be that alterations in amniotic cavitation could contribute to pregnancy loss at implantation. And actually, we know from studies in the mouse embryo that that could be the case. So if we use knockout embryos where the amniotic cavity is not formed, the embryos cannot progress with development, and then they are lost at the time of implantation. So this protein, beta-1 integrin that I just mentioned, in the mouse, when we remove it, we don't have formation of the amniotic cavity. This is a protein that sits in the basal side of the cells in contact with the extracellular matrix, and it gives directionality to the cell. So the cells know where to form this luminal cavity. And in the absence of this protein, this process cannot happen. The cells just don't know where to put this amniotic cavity. So we thought, that it would be interesting to know whether in human embryos this mechanism is also active and whether human embryos are also expressing these integrin proteins at the time of implantation. So this is a zoom-in in one of these in vitro culture embryos. This is the epiblast tissue that you see here labeled in yellow by OC4. And I hope you can appreciate that this epiblast tissue is surrounded by all this beautiful signal of integrin, suggesting that in the human embryo, like in the mouse, integrin-mediated lumen formation may be a very important mechanism at the time of implantation. But is this the only thing that is needed to make an amniotic cavity? Well, we thought there will be something else that is needed, and this is when we started to look at the pluripotency features of these cells. So these embryonic cells that are undergoing this transformation at implantation are pluripotent, meaning that they will give rise to all the cell types in our body. But we know that as soon as the embryo implants, they start to lose this property, these pluripotency features. They start to lose pluripotency in the route towards lineage specification and differentiation at gastrulation. So the hypothesis was that maybe this loss of pluripotency was necessary to form the amniotic cavity and for the cells to become epithelial. In other words, is this pluripotency network of the embryonic tissue controlling amniotic cavity formation? So what I'm showing you here is a blastocyst just as a control. And we can label this pluripotency network by staining for KLF17. You can see we have in the inner cell mass these KLF17 positive cells. So the cells are truly pluripotent. But now we look later on at day 10, and the signal of KLF17 is lost, indicating that the epiblast tissue is progressing in the development, is losing these pluripotent characteristics. But now we can do functional studies and we can do a trick. We know from the work done in human embryonic stem cells that if we add these compounds, we block this loss of pluripotency. So that's what we did. We added those compounds to the human embryo, and now you can see that the expression of those pluripotency genes is maintained and the amniotic cavity is not formed. So these results are really indicating that the epiblast cells need to lose these pluripotent characteristics for the amniotic cavity to be formed. And just briefly, I want to tell you that we can also study these processes in vitro. So we can look at cells that lose pluripotency, cells that maintain pluripotency, and you can see that the human cells that maintain pluripotency, they don't form these cavities. And again, this is conserved in the mouse. So in the mouse, it's the same. When the cells become ready to differentiate, they form these cavities, but not when they maintain these pluripotent characteristics. And we were able to also determine what is the molecular machinery in the control of this cavitation. So we observed that the transcription factor OCT4 plays a critical role in this process. So I'm not going to describe any of the molecular mechanism, but this was the summary of our results, just to tell you that these epiblast cells that we find in the blastocyst, they really need to lose this pluripotent character in order for the amniotic cavity to be formed. Otherwise, amniotic cavitation doesn't happen, and this could lead to pregnancy loss. So in summary of this part, amniotic cavity formation at implantation is fundamental for developmental progression, and this process is directed both by integrins, but also by changes in pluripotency. So by a loss of pluripotency. And we can anticipate that mutations in any of these machineries would lead to post-implantation failure. But I promise a bit of genetics, and this comes now. So aneuploidy, we know, is one of the major limitations of assisted reproduction. So we decided to address how a specific chromosomal alteration could affect amniotic cavity formation and developmental progression. So the approach that we follow was to culture in vitro human embryos with a specific aneuploidies, a specific monosomies, or a specific trisomies. And the question we were asking was, do embryos with different chromosomal abnormalities show different developmental potential in vitro? And just to say that the genetic result is, of course, based on the PGTA. So now I'm showing you here as an example a trisomy 16 embryo, and this embryo developed very nicely. So we can see the embryonic tissue labeled by OX4 here in green. We have also the hypoblast tissue labeled by red, and the embryo just started to form the amniotic cavity. And then we have all the trophoblast around. But then when we looked at monosomies, and this would be a monosomy 21 embryo, you can see that we don't have epiblast anymore. The embryo lost the epiblast tissue, and there is a lot of cell death. So we have quantified these results, and what we did here was to pull together all the monosomies and pull together all the trisomies, and you can see that monosomic embryos present many cases of embryos that die or arrest in development, and I think this is expected because a monosomy would be expected to be much more detrimental for embryo development than a trisomy. And you can see that trisomies do much better, so here 23 embryos just develop normally in our in vitro culture method. But then we decided to focus in this population of embryos, the embryos that both in the monosomy and the trisomy group, they made it. The ones that had all the lineages and they had the proper morphogenetic transformations. And then we quantify more in detail these embryos. So you can see that in the monosomies, even just by looking at the embryos that did make it, the number of cells is decreased. So so we have even doubled the number of cells in the trisomy embryos. But what was more surprising is that when we look at the ICM-TE ratio, in these monosomy embryos that made it, the ratio was higher, indicating that the cells of the ICM in these monosomy embryos were growing much more than in the trisomy embryos. Now, why is this happening? Why in a monosomy embryo, the ICM would have a competitive advantage? So this is a question we are trying to address now. I will leave it as a question mark, but a possible hypothesis was that maybe this population of embryos were not really fully aneuploid and that the ICM was euploid. So to conclude, we've seen that monosomy embryos die or arrest in culture significantly more than trisomy embryos. And the monosomy embryos that do develop up to day 9 are slightly smaller, and they have an altered ICM-TE ratio compared to trisomy embryos. And this also shows that our in vitro culture method is a bona fide platform to assess developmental competency and to assess how specific genetic alterations can affect post-implantation development. And just to thank all the people involved in this work, the human embryo team, of course, Magda, my supervisor, for being such a great and passionate scientist, and Matteo, who is involved, past members of the team, all our clinical collaborators, especially Simon, who is here, and he has been instrumental for this work, Graham Barton at the Center for Trophoblast Research in Cambridge, collaborators for imaging and collaborators for sequencing that I didn't have time to mention, and thank you all for your attention.
Marta.